So, this sun is continuing to fuse together the elements on it into bigger and larger elements through the forces of gravity. And it continues to shine, nuclear radiation. And eventually, it will form iron. And the iron has a very profound effect on the sun, and it causes it to begin to, well, first it will expand very large. And when it starts to form iron, it's starting to run out of hydrogen fuel to fuse together to make helium. And it's starting to run out of helium. So the amount of explosive force begins to wane because it's running out of the basic fuel, hydrogen. And the presence of iron leads to an expansion of the sun, and it expands out usually to fill the whole solar system. And then it continues and continues running out of fuel, and then it doesn't have enough explosive force. And then it collapses all in on itself in what they call a catastrophic gravitational collapse. So you take all that matter that's in the sun, it may have lost some energy, and it may have lost a little matter, but there's still a ton of protons, neutrons, and electrons in there, organized as a dozen or two of those elements. And all those elements come crashing in as the gravity takes over. It runs out of fuel, the explosion slows down, and it begins to collapse on itself. And then all that matter gets collapsed down into a very small point, and then a tremendous amount of fusion takes place in that gravitational collapse. And it squeezes many of the remaining atoms together, and it makes all the atoms in that periodic chart, with the exception of a few that we've made. And then it goes boom in a big explosion. And that explosion, anybody know what it's called when a star explodes at the end of its life? Supernova. Well, this is going to require skill far beyond mine. No, it doesn't. And there's a actual photograph, probably from Hubble spacecraft telescope, and it's an exploded star. It's the results of a supernova. So you've got in this cloud of gas and particles, you've got all the elements. Not only that we need to sustain life, but all the elements that we so far discovered. And what is a star? Well, it's a mass of hydrogen undergoing a tremendous gravity compression and fusing hydrogen into helium and giving off nuclear radiation. What's a supernova? We just discussed it. Why do stars go supernova? They run out of fuel and then they collapse. What happens in a catastrophic gravitational collapse that results in the formation of all the elements shown below? Well, what happens there are massive fusion reactions. And I keep looking over there at the periodic chart, so I put one on there. These are all the elements. Now, with fats, carbohydrates, proteins, and DNA, you only need hydrogen, carbon, nitrogen, oxygen, a little phosphorus, a little sulfur, and that's about it. So we don't have to worry about all of these. We just have to worry about those elements that occur in fats, carbohydrates, and proteins. Now there's also some elements like tin, vanadium, chromium, manganese, iron, cobalt, nickel, copper, zinc that we need to have in very, very small quantities. These are the things that you often get in your vitamin pill, the trace elements and minerals. Some of them, like sodium and magnesium and potassium, function as electrolytes, and they'll be critical in nerve transmission and muscle contraction. So there'll be probably 25 of these elements that we'll discuss. Now, what is the difference between an atom and an element? And you don't need to know, because you already do know, and I'm going to prove it to you. How many atoms of the element hydrogen are in a water molecule? 2. How many atoms of the element oxygen are in a water molecule? 1. How many total atoms are there in a water molecule? 3. Okay. Well, then you know enough to where you'll do fine in this course. Well, this supernova that exploded has enough hydrogen in it that it'll collapse and form a new star, and some of the matter around the supernova outer edges will begin to collapse through gravity and form planets. And these solar systems, we think, evolved like this, and we found a few that are in various stages of evolution. And there's a collision phase of a solar system formation where all the big rocks are running into each other for over billions and billions of years, and eventually most of them get clumped together into what we call planets that orbit the sun. And in this shot, all of them have gotten clumped. Most of the matter's gotten clumped together. You know, there's some asteroids running around out there that like to slam into planets periodically and cause problems. But you know, it's not something that happens every day. Although we do have meteorites hitting the planet every day, but they're usually small, we hope. And some of these planets, in our case one, is just the right distance away from the sun to have liquid water. And liquid water is important for a lot of reasons, but the two biggies are, one, for life to evolve, it's good to have a relatively stable temperature. You know, I go to the desert a good bit and camp and hike, and in the daytime it'll be 75 or 80. At nighttime it'll go down below freezing. So you'll have 60, 70 degree swing in temperature just between whether it's day or night. You stand in the shadows and stand in the sun, there may be a 15, 20 degree change in temperature. It's because the air is so dry. There's not a lot of moisture there. We're here in Florida, we never have a 60 degree swing from morning to night. Maybe when a cold front comes through, it'll get 40 degrees, but because we have a lot of humidity here. So liquid water moderates temperature extremes, swings, where that does not happen on Mars, or it doesn't happen on the moon. You stand in the sun on the moon and you get real hot. You have to have an air conditioner on your back. And you step over in the shadow of the lunar module, well, you need a heater because it's dropped a tremendous amount. So liquid water stabilizes the temperature and allows life to fairly easily come about in ways that we talk about extensively in BSc 1011, which is the second semester. The other factor that water, the other important factor about water is that water is the solvent for all these chemicals. All these molecules and ions have to be dissolved in water for life to spring forth. A good friend of mine is a cattleman, and he buys freeze-dried sperm from Brahma bulls from South America right now. And when the sperm arrives, it's in little Ziploc bags, and it just looks like a grayish powder. And I go over and help him with his cows from time to time, and you open that package up the freeze-dried sperm into a beaker, and you add a little warm water and swirl it around, let it sit there, and you do that a while, and you get a bunch of beakers. And then you pick one up and put a drop under the microscope. Oh, they're all wiggling. And before you add the water, you can look at the little crystals under the microscope, and it just looks like powder. Every once in a while you see an old bent tail or something, you know, sticking out over here, but it's all. But once you add the water, all those chemicals go into solution, and boom, voila, this stuff we call life begins to emerge. And in this course, we're going to distill everything wonderful about life down to chemical reactions. It's called reductionism. T.S. Eliot wrote a poem one time. It was kind of critical of reductionism. He said, as long as I have eyes to see and a nose to smell and a heart to feel, what do I care if some one-eyed son of a bitch figures out a way to measure spring? Back then, microscopes only had one eyepiece you looked at. And so life is not a bunch of chemical reactions. It's something that emerges from those chemical reactions. You know, love is not 2 parts endorphin, 1 part dopamine, and 3 parts serotonin hitting the receptors in your brain. But when that happens in that mixture, it creates this sensation for us that we call love. If you want to have kind of orgasmic love, you squirt in a little adrenaline. And you know, it almost feels, you know, I see the picture of my grandson and granddaughter pop up on my screensaver. And, you know, I look and I can feel that little rush of warm fuzzy, you know. 2 parts endorphin, 1 part dopamine, 3 parts serotonin. So. There's Earth. And these are the, I don't know if you can see them, oxygen, carbon. If you weigh the human body, about 65% of the weight is oxygen, 18% is carbon, 10% is hydrogen, 3% is nitrogen, 1.5% is calcium, and then you start getting down in 0.2, 0.1. So that's just an illustration of most of the chemistry that we're going to study is going to be made up by the top 4, 5, or 6 of those elements. And I ask that you go and find these elements and circle them on the periodic chart. In Chapter 2, we'll talk about the periodic chart in more detail, how it's organized and stuff. All right. Now, 65% of the body may be oxygen, but those oxygen atoms, for the most part, are contained in fats, carbohydrates, and proteins. These are large molecules. Water, H2O, only has 3 atoms. A sugar will have 24 atoms. And a starch, a bunch of sugars strung together in a long chain, will have millions of atoms. So these are big starches and fats and proteins are very, very large molecules. Billions, thousands to billions of atoms. There's some molecules like an endorphin, which is only 10 or 12 amino acids long. And so it may only have 500 or 600 atoms. But most of the fats, carbohydrates, these are large molecules. Water, H2O, only has 3 atoms. A sugar will have 24 atoms. And a starch, a bunch of sugars strung together in a long chain, will have millions of atoms. So these are big starches and fats and proteins are very, very large molecules. Billions, thousands to billions of atoms. There's some molecules like an endorphin, which is only 10 or 12 amino acids long. And so it may only have 500 or 600 atoms. But most of the fats, carbohydrates, and proteins are big. All right, we all know what a carbohydrate is when we go to the store. We're going to talk about what a carbohydrate is chemically. Carbohydrates are made up of chains of sugar molecules. Sugar is a carbohydrate. You hook sugars together in a long chain like pearls on a necklace or boxcars in a train, and you call the whole necklace a starch or a polysaccharide. What's a monosaccharide? One sugar. What's a disaccharide? Two sugars. What's a polysaccharide? Many sugars. And many of the starches that we eat are thousands to 10,000 sugars long. So they get very, very large. The individual sugars are soluble. You know that because you put it in your tea and stir it up, and it dissolves. And the liquid is clear, at least if you use clear water and put sugar in it and stir it up. Once it's all dissolved, you can't see the sugar because its individual sugar molecules no longer crystals. And this glucose molecule has, we're going to write its chemical formula there. Carbons are 1, 2, 3, 4, 5, 6. So carbon, you put a 6 there. Hydrogens are how many? 12. Usually in carbohydrates, the carbons are half the number of hydrogens. And the oxygens are how many? 6. C6H12O6 is a general formula for a 6-carbon sugar. And fructose is what? How many carbons? 6. How many hydrogens? 12. And how many oxygens? 6. So it's the same chemical formula, isn't it? But it's fructose. You find that in fruits. Glucose you find in sugar cane and other places. In the stalks. So they're different. They even taste different. The sweet that you get in the jelly is the fructose sweet. And but what's the difference between the two molecules? They taste differently and they occur different. Molecularly, what's the difference between them? They're the same atoms, but
when they're bonded together?
The way they're hooked together. This one has a different arrangement. This carbon is double bonded to an oxygen, etc. So it's a different structural arrangement. And that gives it a different shape. And shape is going to be very important in terms of how these molecules react with each other and with other molecules. And shape infers different properties on molecules. So those are the 2 sugars, and there's probably a dozen that, maybe 5 that we'll talk about, and maybe a dozen that we'll see. Now these sugars have a capacity to form rings. One of the carbons over here will reach around and bond to another oxygen up here and result in a ring structure of the sugar. It's the same sugar. It reacts the same, whether it's linear or in a ring form. It tastes the same. It's just in a glass of water, if you dissolve these things, they tell me that 95% of them are rings and 5% of them may be linear form. So they can go back and forth, linear to ring form back to linear form. When you draw them as chains of sugars, they're almost always shown in the ring form. And in our textbook, all these sugars will be colored green as an aid to allowing us to recognize them. So we call these polysaccharides. What does poly mean? Many. Like polyester, many ester molecules. Polygamous, many wives or husbands. and polysaccharides are insoluble. Now, let me give you a little exercise about how polysaccharides and monosaccharides are going to play in this course. What do you think this child is doing? Injecting insulin. And what disease does she likely have? Diabetes. And I'm going to raise this. I know you get a little glow there, but those of you in the front row, your job is to never let me write unless I have raised the screen. Though George may be my friend, this is his physics lab, and if I write on the screen, I will have some explaining to do. All right, if you eat a bunch of sugar, this is going to be our blood supply. That box represents all the blood, tissue, and fluid flowing around in your body, inside your heart and veins and arteries. And this box is going to be the pancreas. This box is going to be the liver. And the blood kind of circulates through the pancreas and the liver and back. Of course, it goes to the brain, goes everywhere. But we're just isolating these two organs and the blood supply. You eat a bunch of sugars, and you have a bunch of dissolved sugars as soon as you absorb them. Monosaccharides, if you will. And if you eat too many sugars, for your and it raises your blood sugar level above the normal, then as the blood goes through the pancreas has cells that have the ability to assay the amount of sugar going by in the blood supply. And if that gets above the normal, it will secrete a chemical into the blood supply called insulin. And what insulin does, through a fairly complicated process, I'm going to simplify it. It takes these sugars and hooks them together into a long chain. And we're going to call it a starch. Sugars are carbohydrates, starches are carbohydrates. And starches are soluble or insoluble? Sugars, are they soluble or insoluble? Soluble. Starches, insoluble. So if you take sugars and hook them together in a long chain inside a cell, they'll stay there. They won't go dissolving and moving around. They'll stay right where you put them. And you can store the sugars in your liver as starch. There's a name for that, but we'll wait till chapter 5 to get to the name of that starch. And let's assume that after insulin does a while, then you get up and you do a lot of work and you burn the dissolving, what's left of the dissolving sugar, and all of a sudden your sugar level is too low. Well, the pancreas will sense that as well, and it will secrete a chemical called glucagon. Glucagon. I can't write and spell at the same time. Another one of my multitasking problems. And I like to say it's my bifocals, but I think it's just mild dyslexia. And what glucagon will do, yes.
Untie sugar.
Untie those sugars. Break these sugars out of the starch, and they'll put them back into solution. All you got to do is break one of those bonds between the two sugars, and the sugars now are soluble. Well, they're going to dissolve, and they'll wind up diffusing wherever there's less sugar. You don't have to tell them where to go. They'll just diffuse out because they're soluble, and they'll diffuse wherever there's less sugar. We'll talk about that in chapter 6 and 7. So that's basic carbohydrate chemistry looked at through a particular process. Now, let's add a term while we're at it. Two terms. Dehydration synthesis is what insulin does. Synthesis, what does synthesis mean? To build up or tear down? Build up, to make, create, form, dehydration synthesis. And it involves the loss of the water from the sugar molecules when you do that. So they call it dehydration synthesis. And it'll be a term that we'll use a lot. Glucagon does the opposite, and it's called hydrolysis, because it puts the water back in, and lysis means to split. So it's using water to put back in like a little hatchet, chunk, put the water back in, and it splits the sugar molecule off of the starch. So insulin does dehydration synthesis and builds up starch molecules. Hydrolysis breaks the starch molecules down into individual sugar molecules. So that's how this will work. We'll be applying these concepts. We're not going to talk, well, we'll talk a little bit about DNA today, but we're not going to talk about lipids at all. We're just going to focus on carbohydrates and then proteins and DNA. So, and lipid we'll discuss in depth some other time. If you buy these and eat them at the store, what are you eating? Protein. And proteins are not made up of sugars, they're made up of little, anybody know? Amino acids. So this is an amino acid. Doesn't look unlike a sugar, except the ratios of the atoms are somewhat different. There's how many carbons? 1, 2, 3, 4, 5, 6 carbons. How many hydrogens? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. How many oxygens? Well, there's not many oxygens. And that's one way you can recognize an amino acid. Doesn't have a lot of oxygens. Usually has 2, sometimes 3. And how many nitrogens does it have? It has 1. And they'll usually have 1, 2, or 3 nitrogens. So the reduction in oxygen in an amino acid and the fact that it does have nitrogen. You will never see nitrogen in a carbohydrate, in a sugar. So if you see nitrogen, you know this could be an amino acid. There's only one other thing it could be, and we'll get to it. So looking for amino acids, you look for nitrogens. Now these are all the amino acids. I know you can't see them, you can't even read them on your handout, but they have names like valine, leucine, isoleucine, phenylalanine, histine, asparagine, proline, threonine, tryptophan, those sorts of things. And you hook them together. I'm drawing them as little balls here. That's one amino acid. That could be this amino acid. Well, the next ball is another amino acid. And you hook them together in different ways. Maybe it's leucine, phenylalanine, tryptophan in one to start one protein off. And in insulin, it may be histine, phenylalanine, lysine, and then tryptophan. So different combinations of amino acids produce different proteins. Insulin is 51 amino acids long. Endorphin is 5 amino acids long. Amylase, an enzyme that digests insoluble starch into soluble sugars so it can be absorbed in the cells lining the stomach, is 77 amino acids long. Do you think digestion in your stomach after you eat a protein or eat a carbohydrate is going to be dehydration synthesis or hydrolysis? hydrolysis. Very good. All right, now to DNA. Everybody recognize the double helix there as a DNA molecule? That's kind of how you see it drawn a lot of times in the newspaper and stuff. And we'll talk about the relationship between a chromosome and a DNA molecule. But the DNA molecule is made up of actually two chains. You know, starches and proteins are made up of single chains, but DNA is made up of two chains of things, which we'll talk about, and they're wrapped around themselves. So that's what they call it, a double helix. A helix is the spiral, and two chains spiraling simultaneously are not unlike the hands, the railings of a spiral staircase. Now, DNA is not made up of sugars, it's not made up of proteins, it's made up of special little molecules called nucleotides. And nucleotides there's 4 of them in DNA. Adenine, guanine, thymine, and cytosine. And we're going to abbreviate them from now on as A's, G's, T's, and C's. And we're not going to, when we get to chapter 16, we'll be looking at these molecules in some depth. But it's these molecules that specify your genetic code. When you inherit DNA from your parents, you're inheriting a specific sequence of A's, T's, G's, and C's. And that will turn around and make you. You know, if I had taken your mother's egg before or after it had been fertilized and popped the nucleus out of it, where I took all your mother's and father's chromosomes out and squirted mine back in that egg and put it back, 9 months later she'd have given birth to a little me. So the genetic code, this ratio and sequence of A's, T's, G's, and C's is rather profound in what it can do. It's kind of hard to get a woman to go along with that, though. I mean, I'm good, but I've talked to my wife about cloning myself, kind of doing that, getting one of her eggs, take the nucleus out, put it back in her. But she has no scientific curiosity. All right. Genes occur down a DNA molecule, and these molecules are very long. They're very small, but they're very long. And on the one DNA molecule, you may have several thousand genes. And every gene does one thing. It makes a protein. So we're going to have a little mantra, or several mantras in here, which are phrases that you'll learn to repeat over and over again. And if you like to sit cross-legged while you do it and transcend to a other place, you're welcome to. But in here, genes make proteins is going to be one of our mantras. So everybody say it.
Genes make proteins.
One more time.
Genes make proteins.
I'm feeling better already. One more.
Genes make proteins.
Okay. We'll have up of those that you'll have. But this will save you a lot of grief. Every time you see gene, you'll say it makes protein. Now, you may not understand what in the world does that mean, but that's not important in a mantra right now. It's just that you can pull that up and it'll help you. It's like finding a flashlight when you're trying to get out of the dark. It'll help you find a thread through whatever you're learning. So genes are to DNA molecules as what are to cassette tapes. Everybody remember those old cassette tapes you used to stick in your dashboard? Every once in a while they'd spit the tape out. Bad sign. What is contained on a cassette tape? Information, but what do we call it when we're listening to it? Music. And each individual, what are the individual types of music? What do we call each? If there's 13 different songs. So genes are carried down the DNA molecule. This may be the first gene, that'll be the second gene. If you'd pull that cassette tape out and run it through the player, you could have gotten to the end of the first song and cut that song out, couldn't you? Then run the tape through another player, and when it got to the end, stop it and take the tape out and cut the second song out, right? We used to do that back before we had ways to manipulate audio files. Oh yeah, you could splice together all the songs that you liked. So genes are carried in 1, 2, 3, 4, 5 down a DNA molecule, just like songs are carried on a cassette tape. Now, what do genes do?
Make protein.
Make proteins. Very good. That's it saying, uh-oh. So genes carry the genetic code of a particular protein. If this is gene number 1 from there, well, here it is, gene here. This gene runs from there to there. It carries a code of A's, T's, G's, and C's. The first one you come to is an A. The second one is a T. The third one's a T. C, G, A, T, G, C, A, T, A, T, A, C, A, G, blah, 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 bang. At the end, you've ended the genetic code of that gene. Now that spelling of A's, T's, G's, and C's tells the cell how to build a particular protein. Just like the digital information on the cassette tape told the speaker how to produce the vibrations associated with a particular song. Different magnetic information on that tape will produce different vibrations and produce a different song. A different sequence of these A's, T's, G's, and C's would produce a different protein. So that's where we're headed. How many different kinds of proteins can be made using chromosome number 1? I think it's about 2,000 different proteins are coded for on chromosome number 1. Maybe more than that. And we'll look at all these chromosomes in more detail. All right. Well, this is what the genetic code looks like for a particular protein. I think this is an enzyme that breaks down carbohydrates. I think it's, well, I don't know what it is. Amylase, probably, but irrelevant. So that's what it is. That's your genetic code. That's the genetic code for amylase. Now, how in the world does that tell the cell how to make a protein? Each 3 letters, like A, T, C, A, T, G, codes for 1 amino acid. So it takes 3 letters to tell the cell which amino acid to start with. Which amino acid comes as the number 2 amino acid. That's TTC. AAT tells the cell what the 3rd amino acid is going to be. CTC. Now I'm going to show you a little dictionary where you can look up what CTC says is the next amino acid. So that's how it's going to work. There are 60 letters in each line. You could count up the lines. But 60 letters would give you would specify how many amino acids. If it takes 3 per amino acid, 20. So that's the idea. What do genes do? They make proteins. How many bases, A's, T's, G's, or C's, does it take to code for 1 amino acid? How many? 3. In DNA, what does A pair with? Well, let's look at this DNA molecule. Now what we've done is we've taken the double helix, I've unwound it and taken an iron and ironed it flat so we can easily see the A's, T's, G's, and C's. And there's one side of it, there's the other side of the DNA molecule. can you see either on yours or on this one what does A always seem to be paired with?
T.
T. How about C? What is it paired with?
G.
And we'll talk about why there are two chains and what's the benefit of that. Now, these codes like ACC, codes for tryptophan. AAA tells the cell the next amino acid is phenylalanine. CCG says it's glycine. AGT codes for serine. So, a chain of sugars is called a polysaccharide, or if we're going to eat it, we call it a starch. A chain of nucleotides, A's, T's, G's, and C's, is called a DNA molecule. Now, as a general class of molecules, these DNA molecules are called nucleic acids. So sugars form starches, which are carbohydrates, and these nucleotides, the A's, T's, G's, and C's, form nucleic acids. All right. Now this is basically how it happens in a cell. You've got a DNA molecule. Now in our cells we have those DNA molecules inside the nucleus. And we inherited all of these from our parents. Half of them we got from mama and half we got from our father. There's another kind of nucleic acid besides DNA, and it's called RNA. And DNA is colored blue in our textbook, and RNA is colored red. So this is a DNA molecule, or maybe that's a gene. Yeah, that would be a gene. This one would be a DNA molecule. This would be an RNA molecule. What do you think the little balls are? Well, the big brown thing is a ribosome. But we got a chain of balls over here, and we've got little balls there floating around singly. Amino acids, very good. And this ribosome gloms onto the RNA molecule and runs down it, kind of like a zipper down a zipper glide, and chink, 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 reads the code and hooks the amino acids together in the right sequence. And then when it finishes, this little protein pops out the other end of the ribosome. So those are the cellular structures that are going to be involved in this process. Now, what is happening? This arrow indicates that the genetic code on the DNA molecule is being copied onto a messenger RNA molecule. There's several different kinds of RNA. We're going to call this RNA little m-RNA for messenger RNA. And it's carrying, what's the message that it's carrying down to the ribosome? The DNA message, the genetic code. And so there's a wrinkle here. DNA is made up of A's, T's, C's, and G's. And RNA, however, is made up of A's, U's, C's, and G's. Now, your cell looks an awful lot like a thiamine molecule when I show you the structure in chapter 16. It's only got one or two atoms different, and they're just different places. But chemists have to give it another name. So it adds a little level of complexity to this thing. But you'll get it real quick. This is the template strand of the DNA that's actually carrying the genetic code. This strand is there just to keep this code in place. And when I make a peanut butter and jelly sandwich for my grandson, I always put the peanut butter on it and the jelly, then I put the top on it. Now mine, I just put the peanut butter on one piece of bread and the jelly, and I eat it over the sink usually. But if I'm going to give it to my grandson, why do I always put a top on it?
To keep it in place so nothing spills out. Stays together.
What's he going to do with it real quick?
Turn it up, eat it, and dump all on it.
Yeah. Drop it on Grandma's carpets, my major fear. And since she brought me a carpet cleaner for Christmas, it's been something I've started to worry about a little more. I guess she means I've got to clean it up. So this part of the second strand is just there to keep the top strand stable. It's less likely to mess up, mutate, change, knock one letter out and replace it with another than if it's got a second strand there stuck to it than it would be if it were just loose and flopping around. So the top strand is carrying the code. We'll call that the template strand. And A's always bond to T's and C's to G's. And the same thing is true for RNA. This A will stick to what would normally be what? A T. But we don't have T's in RNA. We have what instead? U. So A goes to U. C would be to G. And these literally, what you do is you rip this bottom piece off temporarily, and these U's and G's and T's actually stick to these molecules, and then they peel off, and that's how they'll be formed. So C would stick to G, A to U, A, U, A, U, C to G, C to G to C, A to U, G to C, and T to A. So we would have successfully copied the genetic code of the gene to a piece of messenger RNA. So we're right here. We've just done that era. Now, we go out and find a ribosome, this brown thing, and it comes along and locks onto the messenger RNA molecule and goes down and reads the genetic code. On the next page, I think, or is it the bottom of that page?
The dictionary?
The dictionary.
Yeah, this page.
Yeah, the bottom of that page. The ribosome is going to see UGG, and what amino acid is it going to use to start the protein with? I can't find it. There's a system, but I never use it. I just look around until I find it. UGG? UGG, I see it. The top, the fourth row from the top all the way over on the right-hand side.
Yeah.
See it? UGG is TRY, that's tryptophan. So write TRY there. Now the ribosome comes to the next three letters, UUU. What amino acid does uracil code for? PHE, phenylalanine. And it makes a little bond between the two. Then the ribosome moves on to the next one, which is GGC. What is that code for? GLY, which is glycine. And UCA? SER, which is serine, and so on and so forth. Now, this process of going from a sequence of A's and T's, or A's and U's and G's and C's, to a sequence of amino acids is called translation. I wonder why would they call it translation?
Translating.
Yeah, isn't that what we do when we go from French to German or English to Spanish? We just take the sequence of the letters and we change them into the sequence for the other language. So it's called a translation step. And we call this step of copying the gene, the genetic code off the DNA onto a piece of messenger RNA, we call transcription. What is a transcript?
Copy.
A copy. If you go to court and there's a court reporter there and he produces a verbatim transcript, it's a copy of what was said. Now let's talk about what a mutation is. If something comes along, like alcohol or tobacco, nicotine, or other chemicals, or sunlight's a biggie, and knocks one of these letters out, A's, T's, G's, or C's, and it's replaced by another letter, then we have what is known as a mutation. And if this was G, 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 and we knocked the G out, it was G, G, A, we popped the A out and put a G in there. So G, A, G would code for one amino acid, and now G, G, G codes for another amino acid. We haven't copied it on the messenger RNA yet, so don't try to look it up there. But we would have changed the first, second, third and fourth amino acids in that gene. All the rest of them would be the same. But the third and fourth one are wrong, are different. It's like a misspelling in a word. So a mutation is just a misspelling in the genetic code, and it results in the wrong amino acid being put in the chain as a protein is assembled. And that makes them different shapes and makes them react differently, and it gives rise to a lot of the diseases that we suffer under because of these genetic mutations. Well, let's show how a mutation can cause a disease. Well, here's a protein being formed, and this is going to be a hemoglobin protein. What does hemoglobin do for you? Carries oxygen in the blood. And hemoglobin, when it has lots of oxygen, is actually a reddish pigment. So here are the amino acids that are hooked together. And each amino acid, which are shown as balls here, showing the individual carbons, hydrogens, and oxygens as well, they will stick to each other by a process known as hydrogen bonding. It's going to be very important. Hydrogen bonding. And they will stick together and form twists and kinks and pleats. And this ribbon in here is this chain of amino acids shown as a ribbon so you can see it spinning. You know, if I had a string of pearls here and I twisted it, you couldn't tell I twisted it by looking at it. You know. But if it's a ribbon and I twist it, you can tell from the back room it's been twisted. So they're shifting from balls in their diagram to ribbons so we can see this twisting and changing. And that's called the secondary structure of the protein, the twists and changes. This is called the primary structure of the protein, sequence of the amino acids. And as a result of the secondary structure, which is formed from amino acids sticking to each other, and serine may stick to tryptophan, but it won't stick to phenylalanine. So if you have a mutation, some of these sticky points get changed around. And as a result, this secondary structure, the twisting and turning, is no longer the same. And it results in an overall change in the three-dimensional shape of the final protein. So we call this primary structure, secondary structure, and tertiary structure, the shape. This is showing both the secondary and the tertiary structure. Now, shape is very important. You know, the endorphin molecule, that feel-good molecule, fits the endorphin receptor like a lock and key. You know, you go down to the hardware store and you have them make you an extra copy of your front door key. Before you pay for it, you hook them together and hold them up and make sure they're perfect. And then you pay for it, you take it home, put it in the lock, and what happens? Doesn't work. It may be close, but it ain't perfect. And this endorphin has to fit the endorphin receptor like a lock and key. Hemoglobin has to fit the oxygen molecule like a lock and key. So if you make a hemoglobin molecule with just slightly the wrong shape, it's just like your front door key. It doesn't work. And unlike the front door key, it won't wear in time. It'll just never work right. So it's a shape phenomenon that is due to the shape phenomenon in the hemoglobin molecule that was due to the wrong amino acid being put in a sequence. I think the hemoglobin molecule is 146 amino acids long, and it's one out of order. One is wrong out of that 146. And that results in enough of a shape difference to where the hemoglobin with that mutation doesn't carry oxygen very well. What do we call that disease? Sickle cell anemia. And this is just an example. It's the 1, 2, 3, 4, 5th, the 6th amino acid. Supposed to be, looks like glycine, and instead it's valine, another amino acid. And the genetic mutation is it was CTT at that location, and the mutation makes it CAT. Now we've covered a lot. I don't expect all of this to be crystal clear until next Tuesday when you come in and you take our first exam. We'll have a chance for a little review. But let's talk a little bit about how sickle cell anemia is inherited. We've looked at the molecule, hemoglobin. We've looked at the gene that was mutated. Well, we normally think of sickle cell being something you inherit, and it is 99.9% of the time. I'm going to use a big S to indicate normal hemoglobin. And I'm going to use a small s to indicate sickle cell hemoglobin. And we have 2 genes for every trait that we have. My mother was blue-eyed. She gave me a blue-eyed gene. My father was brown-eyed. He gave me a brown-eyed gene. And my eyes are what color? Brown. But I'm carrying what gene in my cells? Blue. A little blue. So I've got a big B, if you will, and a little b. The big B for the brown and the blue. How come I've got a big B and a little b, but my eyes are brown? It's dominant. Brown is dominant over blue. We'll talk about how it's dominant in a minute. Well, normal hemoglobin is dominant over sickle cell hemoglobin. So if I've got big S, I say I'm normal. I have 2 functioning hemoglobin genes. If I've got a big S, little s, say my mother was carrying a little s, she would be a carrier, she'd had the sickle cell trait. She would be, I would be normal, but I'm a carrier, right? Mm-hmm. Or I could be little s, and I'd have sickle cell anemia. So that's how it works. And then we're going to do 2 chapters on genetics and focusing largely on genetic diseases, but we'll look at some normal genes as well. And this pattern is going to repeat over and over again. But let's assume that I'm a carrier, and I marry a woman who is a carrier. Well, what's the chance of our first child having sickle cell anemia? 50-50. How much? 50-50? 25%? 24%? 15%? Anybody 15%? Well, let's see. These genes, the 2 genes that you have, always come apart when you make a sperm cell or an egg cell. So let's assume she's making some egg cells. She can either put a big S in an egg. The next egg comes along, she'll put a little s in it. Now, when he makes the sperm cells, the first one gets a big S, let's say. and the second one gets a little s. Well, what are all the combinations that can take place? Well, the big S could fertilize the big S. What would that be?
Normal.
Normal, big S, big S. The big S could fertilize a little s, and what would that be?
Still normal.
Still normal, but a carrier?
Carrier.
The little s could fertilize the big S. What would that be?
Carrier, but normal.
Carrier, but normal, little s, big S. And the little s sperm could fertilize a little s egg, and that would be what?
Mutated.
A mutated sickle cell. So that's how many individuals are going to have sickle cell out of the total.
1 out of 4.
1 out of 4, or 25%. There's a way to make a little table, and you put the eggs up here and the sperm over here, and then you can kind of do this, and it makes it a little easier to see. But that's the idea. So the gene of this little s, it's indicated by that little s, is a mutated gene, which causes the wrong amino acid to be in place, which causes the protein to function either not at all or much less well. All right. We will also look at a few things other than genetics. We're going to look at how the cells operate, burn fuel, and convert the fuel into usable energy in the cell, and some other processes. But the lion's share of the course is going to be spent on genetics. The chemistry is first, the cell machinery is second, and then we look at how genes make proteins for the remainder of the course. Now, on next Tuesday, you'll come in, we'll do a little review, and the exam I'm going to give you is going to be very short, and it's going to be over this material. You don't have to look up anything in the book. You know, if something's unclear, you can go on the internet or stuff, but I just want you to understand what we've covered, and I want you to get used to taking tests. Now, the purpose of doing this is what? Doing this kind of overview is what?